Hi, I'm Dr. Pam Blundell from the Institute of Psychological Sciences at the University of Leeds, and I'm going to be telling you today about Pavlovian conditioning. I'm going to outline for you some of the principles of Pavlovian conditioning, and then I'm going to tell you about a particular example of how Pavlovian conditioning has been applied to solving a problem in humans. I'm going to start by telling you about Pavlovian conditioning, and then I'm going to give you an example of how it's been really useful to help us understand a human problem of anticipatory nausea and vomiting. So everybody knows about Pavlov and Pavlov's dogs. The basic principle is that if you take a neutral stimulus, such as a tone or a bell, and pair it with a biologically significant stimulus, such as food or an electric shock, then you get a change in the behaviour to that previously neutral stimulus, which is appropriate to that biologically relevant stimulus. So in Pavlov's case, he had metronomes and lights, it wasn't all just bells, and he paired those with food. And he looked at how much dogs salivated. He literally counted the drops of salivation that they made. And if you go and get his book, available from all good bookshops, you can see his tabulated tables of how many drops of saliva they gave in each of the different conditions of the sorts of experiments that he ran. Everything that I'm going to tell you about today comes from Pavlov's work. Initially, there's no response to the neutral stimulus. Why would there be? It's neutral. There may perhaps be an unconditioned response, a slight flinching if there's a tone, or rearing if there's a light, but that very quickly habituates. As trials proceed between the CS, the neutral stimulus, and the US, the biologically relevant stimulus, the behaviour of the animal changes. The animal begins to change its behaviour during that CS, that previously neutral stimulus takes on some new meaning. The greatest learning occurs during the first few trials, and here you can see a typical learning curve. So initially animals show no conditioned response, and then over time the conditioned response increases with the greatest learning during the first few trials. Then learning reaches an asymptote. This is where the curve flattens off and no more learning is possible. Through Pavlovian conditioning, animals come to learn about features of their environment and their predictive qualities. This is really useful for animals if they can learn what's likely to happen next and they can make appropriate responses to that. So to recap, the elements of Pavlovian conditioning. There is the unconditioned stimulus. This is the biologically relevant stimulus which is presented, such as food or an electric shock. There is the unconditioned response. This is the animal's normal, natural, reflexive reaction to that unconditioned stimulus. It might be salivation or approach to the food. It may be freezing or fear to a tone. There is the conditioned stimulus, that previously neutral stimulus such as a tone or a light. And then there is the conditioned response. That is the response that the animal comes to make to the conditioned stimulus over multiple training trials. In Pavlovian conditioning, we can study a number of different species, from humans, chimps, dogs, cats, slugs, snails, fruit fly. All species show some amount of Pavlovian conditioning. Indeed, there have been debates whether plants show Pavlovian conditioning. As such, Pavlovian conditioning is obviously very, very important and of fundamental significance for animals. I'm now going to give you a couple of examples of Pavlovian conditioning that are used in animal research before I go on and show you how it can be used to help humans. One of the classic examples of Pavlovian conditioning is the rabbit nictating membrane or eyeblink response. Rabbits have effectively got two different eyelids, and the second eyelid or the nictating membrane will respond to threats such as electric shock or an air puff. If a tone is paired with an air puff or mild electric shock, then over time, in fact very rapidly, that tone will come to elicit an eye blink. And this form of learning is an excellent way of studying conditioning within the rabbit. Here you can see some graphs which show the results of an eye blink conditioning experiment. On the left you can see a graph which shows the acquisition of the eye blink, and it shows the percentage of trials that have got the eye blink. And very rapidly, 100% of trials are showing a conditioned eye blink response. On the right, you can see a generalisation curve. So a tone was paired with the electric shock, and then different tones were paired to the rabbit. and we can see how well the response, the conditioned response, generalises to different tones. And we can see that the optimal response is the tone frequency that the rabbits were trained on, and as you get further away from that frequency, then the number of responses reduces. This shows generalisation to similar tones and discrimination with different tones. Another form of conditioning which is often studied is taste aversion learning. Now, many of you will have some taste aversion yourself. You will have had an experience where you've eaten a novel food and then you've subsequently been ill, and then you can't eat that food anymore. Many people have got taste aversions. Lots of people have a taste aversion to whiskey. I've got a taste aversion to banana milkshake. But very few people have got a taste aversion to beer, and we'll get onto that towards the end of this podcast. In studying it with animals, we give the animals a novel flavour, and then we make them feel ill by giving them an injection of lithium chloride. So with rats, we can study taste aversion learning by giving them a novel flavour and then giving them an injection of lithium chloride. This makes the animals feel ill, but because rats don't have a vomit reflex, we can't ask them how ill they are, and so we have to assess their illness by seeing how much they will drink of another drink. So the typical experiment, a rat would be given a drink of vanilla, followed by a lithium chloride injection. On the next day, the rats might be given a drink of almond with no lithium chloride injection, and then we can look and see which of the drinks they will consume. Of course, we counterbalance between our animals, so half of the animals will have an aversion conditioned to almond and half will have an aversion conditioned to vanilla. So what relevance has this got for human cognition? There is a condition called anticipatory nausea and vomiting, which affects 29% of cancer patients. 29% of cancer patients will feel anticipatory nausea before they go and get chemotherapy. 11% will have vomiting before they go and get chemotherapy. And it's fairly clear how this can be accounted for by Pavlovian conditioning. The CS is the hospital environment. The hospital is previously neutral and comes over time to evoke nausea response. The US, the unconditioned stimulus, is the injection of chemotherapy. The unconditioned response is the nausea which is felt as a result of the chemotherapy, and the conditioned response is the anticipatory nausea and vomiting. Now, this is a really serious problem because when patients suffer from anticipatory nausea and vomiting, it can actually prevent them from having their chemotherapy, and it certainly makes it very unpleasant for them. In order to treat anticipatory nausea and vomiting, then, we need to have a good understanding of Pavlovian conditioning. Here we have an experiment which was conducted, which looks at anticipatory nausea and vomiting and the role of classical conditioning. In this experiment, women who were undergoing chemotherapy for breast cancer were given a novel drink before the chemotherapy. So this novel drink acts as a CS. They then had the chemotherapy, and what was assessed was the nausea response to that drink. So now we're actually studying within an experiment the effect of the chemotherapy on the aversion to the novel drink, to establish that this is a plausible model for the cause of anticipatory nausea and vomiting. On the right, you can see the experiment group and the control group responses. You can see their ratings of nausea. The experiment group, before they had their drink, which is the white bars, showed very little nausea. But after they were given the drink, they showed a lot of nausea. So the experimental group, those that had the novel drink before the chemotherapy, are showing a large nausea response to that drink. This demonstrates that it's plausible that Pavlovian conditioning is responsible for the anticipatory nausea and vomiting. The control group, who simply had the drink at a different time from the chemotherapy, do not acquire that nausea response to the drink.